Thank you so much, Doctor, for that wonderful introduction for both myself and the topic of the day. Now, of course, we have heard so much of autoantibodies from morning, and still there's a lot more to learn. But what I would like to take you is through a wonderful journey of how the techniques have developed and where we are. Because when it comes to autoantibodies, why the topic today is basically because we are currently now at the brink or the threshold wherein we know that from basic immunology, we are moving to precision medicine. And these autoantibodies do contribute in a big way. So why is it that they matter? Because autoantibodies are useful for early diagnosis. It has been noted that there are some antibodies which are present much before the disease manifests. For instance, anti-CCP is known to be present much earlier before the disease manifests, as well as more than that, to earlier diagnosis. If a patient has a dsDNA antibody, then you know that there is a likelihood this patient might go in for a renal involvement, so we need to treat it properly. So earlier diagnosis before the organs are damaged, prognosis prediction, because specific antibody profiles predict organ involvement, which I have already said, as well as, for instance, if you have a positivity of an MDA5 in a dermatomyositis cases, then you know that this is going to be a severe disease with lung involved, and the way you treat would be very different from a patient who has other mild antibodies which are present in a case of a myositis. So then it comes to serotype guided therapy, biologic selection, disease monitoring, particularly a few antibodies. Most of the antibodies do not really help in disease monitoring, but there are few antibodies like dsDNA, the PR3 antibody which is there in vasculitis, as well as anti-CCP, which sometimes help in tracking the disease activity. And of course, the earlier you diagnose, the better the treatment. Before organ involvement, before failure sets in, you know that you are saving the cost of the patient. So when we are having this, what are the key diagnostic challenges? Why so much on autoantibodies when we are able to do diagnostics so easily with other conditions? The problem is the limited specificity. Because autoantibodies are present in multiple autoimmune. A specific antibody is not necessarily present only in one condition. It is present in multiple autoimmune disorders. So that is the problem, because for instance, you can have a Jovan, a positivity of a Rho52 or a positivity of a SSA, SSB in lupus, in Sjogren's, in other conditions. ANA can be positive in autoimmune as well as so many other conditions of autoimmune disorders, not just that, even infection, malignancy, and sometimes in normal individuals. Sensitivity. Not all patients with disease have detectable antibodies. So if most of the time, if an ANA is positive or an autoantibody is positive, then patient is having the, you start working up. But just because it is negative, it does not mean, because some of them they may present later, and you need to look at the other clinical features. So the limited sensitivity which is there, variability between the individuals. A lupus in one patient may have different antibodies from another patient. And not just that, even in the same patient, if you really look in a follow up the patients, the antibodies profiles might change. And of course, the lack of standardization and reference ranges as far as the lab is concerned. So what happened? So when we look at the autoantibodies, when you look at the earlier methods were the counter immunoelectrophoresis as well as double immunodeficiency, immunoprecipitation assays, which basically depended on the precipitation reactions or the antigen-antibody reactions, where you use soluble antigens and you look for the precipitation line. And here, of course, they were very highly specific and used native antigens. The next came but these took a really, really long time, and some of them even took weeks for the results to be available. Then came the conventional methods, which we are currently using. That is the indirect immunofluorescence method, which is very commonly, we have heard about it from morning, which was introduced in 1950. The ELISAs, specific assays, line immunoassays, which have a more sensitivity, as well as they are faster to do. And then now we are looking at the new emerging techniques like the cell-based assays, the luciferase immunoprecipitation assays, cytobid assays, microarrays, which are multiplex assays, which are AI integration into them. So when you look at the immunodeficient techniques, they had their advantage, of course, as I've already said, they are precipitation reactions. And the good part over there is they use native antigen. So it has given us a lot of contribution like SM antibody, which was actually detected by the counter immunoelectrophoresis. For that matter, the ELISAs do not have the specificity. They are so very specific, that is why they were included into the criteria of lupus, because they were seen only in counter immunoelectrophoresis and the immunodeficiency techniques. But as I said, though highly specific, challenges in the testing. Then came the, when it comes to immunoprecipitation assays, to date, we have detected so many antibodies in systemic sclerosis, myositis and all. But when it comes to systemic sclerosis, here, it is still the gold standard for identification of antibodies in systemic sclerosis patients. Here you use polyacrylamide gel analysis, electrophoresis is done, and then you look at the identification of these antibodies, very high sensitivity, specificity, and identifies ribonucleoprotein antigen. So why in systemic sclerosis? Because most of the antibodies which are there in systemic sclerosis, they are ribonucleoprotein antibodies. And these antibodies, they work better only when they are in the native conformational form, in the native nature and in the, so when you do the purifications, sometimes you lose this conformation. And that is how sometimes you do not detect them. Once detected, now of course there are methods where they have identified that you can still line them on the nitrocellulose membrane, or, but to detect new antibodies and when there is doubt, still it is being used in very research labs, not really used in the diagnostic. The most common method which now we use is the indirect immunofluorescence method, a well-established technique for a detection of a wide variety of antigens in tissues, in cells, by the use of specific labeled antibodies, where, this is the count, yeah, specific label antibodies. So here basically we use slides on which the antigen is coated, which is generally the HEP2 cells, to which a primary antibody is added, and then a secondary antibody which is tagged with the FITC. What is the problem over here is that we require an immunofluorescence microscope, as well as it needs expertise for reporting this assay. The ANA, when you detect, so where do you use? Basically ANA, it is of course the gold standard method for detection for immunofluorescence. It's also useful in ANCA as a dsDNA confirmatory assay. So it's the most specific when you do by immunofluorescence, that is the Crithidia lucile immunoassay. Anti-mitochondrial, anti-smooth muscle antibodies, which are used in primary biliary cirrhosis, autoimmune hepatitis workup. So when it comes to ANA, when you look at, this is not a single antibody. It is a heterogeneous group of antibody which is directed against the different nuclear as well as the cellular antigens. So now actually they say it's a misnomer, because we know that it is the antibodies are present not just in the nucleus but also in the, and about cytoplasm, and about 130 to 150 antigens are noted. So though we have identified currently about 31 with their patterns, but there are more, about 130 to 150 antigens are already there, where we have labeled and disease association has been given. Various patterns are seen which will clinically characterize in helping us what could be the possible antigen depending on the pattern that we see, and then what could be the disease association. Only thing is, as I said, it needs expertise and it had a low throughput. So that is why a lot of ELISAs also came into the picture. But however, when I will come to that discussion again. But so when you look at the ANA under the immunofluorescence, you have this is the current guidelines, that is the international consensus for reporting of ANA patterns, where you have a negative. If it is positive, you say whether it's nuclear, cytoplasmic, mitotic, and we still have some unclassified. Another 3 are already been detected. This is as of 2023. This was actually done in 2014 in Sao Paulo. They had come together to bring in a harmonization for reporting of the ANA patterns. Why it is important? Very well told by Dr. Ashish in the morning. If it is homogeneous, you might see that there is a specific antigen association and a disease association. So it becomes very important that ANA should be done by an immunofluorescence method. I will not go into this crowded slide completely. He has covered it quite well actually. So, but then there were challenges. It was slow, it was time consuming. And not just that, as mentioned by Dr. Somyashi, that Sometimes some antibodies like anti-Rho antibodies were not detected on the Hep2 cells. So what happened was genetic technology came into the picture and we could develop specific substrates and use knockout cell lines with Rho60 which improvised from Hep2 to Hep2000. Then people started adopting Hep2000 or Hep2010, so we do not miss out on the Rho60 antibodies. And now a knockout substrate antigen has been brought in for DFS, the dense fine speckled pattern, the DFS antigen 70. Not just that, the challenge of having slides, manual processing, and also having the concern of not able to store the results on the system was a concern with IF. So now we have automation, and you can, these automated system, they identify, store, and analyze the pattern, and you can report based on the fluorescent intensity. We are now able to interpret the titers also of the antibody. So that is what we are doing, and this brings in a standardization as well as a TAT. So this is the instrument that we are currently now started using, where we do the processing on a processor, and then later on when you load the slide over here, which has the laser inbuilt, you can actually see the pattern over here. This is the slide, and each well is analyzed, and you can save images and you can interface it to your lab software. So that is the good part, and this brings in harmonization. Though I will tell you very clearly, there are still a few challenges because this is such a evolving area, and every day we are getting new patterns and new things. These instruments are through machine learning and through deep learning, depending on the analysis of the patterns, but still there are areas where we need to still review the slide regularly because some of the patterns are not identified and we might miss on them. But still it brings in a lot of standardization and you can store data. Then came in the era, once you have the ANA positivity, then you wanted specific assays and what you could do and quantifiable assays, so came in the ELISA, which is an enzyme-linked immunosorbent assay, where the wells are coated with the antigen or antibody of interest, and then you add the patient sample to which you add the enzyme tagged antibody and look at it, measure it in the colorimeter. So various ELISA panels are available to measure the antibodies against the specific antigens, particularly when it comes to lupus, where you have anti-dsDNA, anti-SM antibodies, SSB, anti-RNP antibodies. Systemic sclerosis also you have centromeres, SCL70, RNA polymerase, myositis, JO1, PL7, Me2, vasculitis and antigen. But when it comes to ELISA, there is again which antigen to choose is very important, what you coat onto the antibody. To give a simple example, when you look at the snRNPs, which you call as the small ribonucleoprotein antibodies, there are about 6 types. And of the 6 types, it is only the SMD which is the most specific for SLE. So when you choose a kit, it becomes important how you choose. When it comes to UNRNP, it has different fractions like A, C, and so when you, at least for it to be significant with lupus, at least 2 antibodies, 2 of those antigens should be coated onto a well. So it becomes very important that both the vendors, companies which provide the kits, they have those, and when we choose a kit as a lab, we should be aware that we should choose these kits. The other challenge is of course the false positives which can happen because of cross-reactive antibodies, basically because of matrix effect. For instance, if it's a patient has a very high rheumatoid factor, then endogenous biotin binding might happen. Due to non-specific binding, anti-dsDNA particularly is known to cause that it will go and bind to these antigens on the well and give you a false positive reaction. So you might come with a patient who doesn't have and is having dsDNA positive. It depends on the assay where we are using. And then it comes to epitope dependence again, because it depends on the linear or non-conformational antibodies will go and bind to it, giving a false positivity. And of course the inter-assay variation between the different manufacturers and as well as the antigens, buffers, detection systems, which all give to different results. Just a simple example, as I said, dsDNA particularly, this is a antibody with lot of interest, basically because many a times in line immunoassays they are not detected. We do the enzyme immunoassay, the ELISA. It is a quantitative assay, you require it for monitoring. But when you do an ELISA on this, earlier antibodies, as I said, they used to sometimes give false positive reactions. So a lot of companies came in with different technologies. So this is one antibody where they try to take only the isotope so that it does not bind to the wells over there, And there was another company which tried to mix it with nucleosome, which had the nucleosome, and so that the dsDNA doesn't bind there, and then you look for the reaction. Definitely specificity wise, both of them were reliable. That is the enzyme linked isotopic assay actually, what I'm seeing here. And this is, which is complexed with NCX, that is the nucleosome. And this showed a superior sensitivity besides the specificity. This was a study which was published in 2023 in Germany. So when I say that when we are looking at the assays, how we interpret what you do, clinical features still takes a very big, are very important when you are interpreting these assays. And if you feel that something is not matching, please give a call and check out with the consultant or to the lab. Now when it comes to chemiluminescence, ELISAs, we need more automation, and everybody wants it to be putting in a biochem instrument which just does testing. You run the QC in the morning and it comes. Definitely it helps a lot, but then there are some chemiluminescence assays which are available, wherein you use a solid phase, which are magnetic particles, and they are coated with the specific antigens, and then you measure this. The chemical reaction is then measured by the luminar, by the fluorescence over there, and you see what is that is the principle which is there in chemiluminescence, which is then again identified by the photomultiplier tubes. This particular assay is of course been very useful in patients with rheumatoid arthritis. Now anti-CCP, I do not think, we started off with ELISA, most of us have moved on to the chemiluminescence. So some assays it definitely helps with very wide sensitivity, and it gives, even microliter, nanoliter also it gives results, so you are able to get good reference ranges, but wide dynamic range allowing accurate measurement across broad concentration. But limitation, most of these chemiluminescence assays are available, are closed systems. So where the company does, you can run only their company products on that. And not all of them have the currently available antibodies for all the diseases that we have seen, which we are otherwise seeing in other platforms like ELISA or by line immunoassays. This is an assay which is very popular, particularly in our country. I know some countries do not recommend that so much, is the line immunoassay. Here basically the antigens, are actually coated on the nitrocellular membrane, and then again it is like an enzyme. You add the sample to which you again detect the antibody by an enzyme reaction, and then look at the band which is detected. So this again allows for a lot of automation. Here this is a processor for the line immunoassay, and two different companies. Here this is a microdot immunoassay, where not just does it even for each sample you have a separate strip, you have a separate well, so there is no mix-ups, no carryover, so those are the advantages, and it has a holder which you just load it into the scanner and you read it visually. So this is how you get it. So for instance in this patient, this is a 25 antigen ANA quantification which is done semi-quantitative, I could say, where you are able to see. Here of course it's a negative sample, but this is a blot, just an example for the blot assays. The advantage is multiplex assays allow simultaneous detection of multiple autoantibodies, objective, semi-quantitative, but again here they have limitations about sensitivity, reproducibility at times, significant interarray because of the various epitopes used, different epitopes which are used by different vendors, and a little bit of a lack of standardization, because now there are quite a few companies in the market which are doing the line immunoassays. And how do you estimate it? That's also what matters. And what antigen have they coated? So let us just look at a few applications, how beautifully it helps us. Sometimes ANA is positive. This is a case of a lupus. RNP is positive, dsDNA, nucleosome. And becomes very easy for this patient then to be diagnosed, as well as to see for which are the antibodies. Once you see dsDNA, definitely you please do a ELISA also. Dr. Ashish has very nicely told how myositis, particularly this particular area, has taken up a lot of development. He has gone up to 2023. This is an old slide that I have put in. And every day you will see that there is a new antibody. I mean, not every day, every couple of years you have a new antibody which is coming in. And earlier, of course, as he said, it was basically antibodies for dermatomyositis like Me2 alpha, beta, SAE antibodies, MDA5, TIF, and XP2. And then for polymyositis, it was SRP, JO1, and then overlap, you had PMSCL, 70 PSCL, 100, and sporadic inclusion, which is the C1A antibody. So this is how it was, but now you do look even in dermatomyositis. Suppose a MDA patient has MDA5, then he will have a more aggressive clinical course. A patient who has necrotizing myositis, you will probably look for SRP presence as well as for looking of the HMG co-reductase. If it is HMG co-reductase, you start thinking in terms of a statin-induced myositis. This could be a mild form where the patient has only some Me2, but still TI, if it's borderline, I wouldn't give much significance to that because it's in the equivocal range. So that is one thing I wanted to highlight. Sometimes when they are in the equivocal range, please look at the test, whether it is really significant over there, and don't just consider it as positive. That was one thing why I put it. So this is for myositis, that's how it helps. This is one particular pattern, for instance, if a ANA you have a centromere pattern and you need not even do a profile for this. It's very clinically correlates very well, this pattern correlates very well with the profiles that we do. And again, diffuse systemic sclerosis, where you have SCL70, so which have more aggressive compared to the limited form, and then here the patient also has RNA polymerase 3 positivity, which indicates. So why these are important is basically because you are able to stratify the case into how would be the prognosis, how to treat them. Better prognostic antibodies are centromere and all, and those which are having worse prognosis are THTOs, SCL70 and RNA, in between being the fibrillary and U3. So this was with the routinely, commonly used in many of the labs which are doing autoimmune assays. But then there are a few areas that when you do ELISA and when you do a blot assay, we are not able to detect the antibodies. And because of the changes which happened in during the purification process. So there we require live cells, so use native antigens on live cells, and which mimics a in vivo situation, and for the conformational antibody, and this has been very useful for detection of neuroimmune disorders, particularly myasthenia gravis, encephalitis, and this can be done both in the serum sample as well as on the CSF. And you can see here, this is a patient which has got LG1 transfected. Here you look at the neurons and glia, take the recombinant cells and culture the primary neuronal cells along with some of the transfected cells and see for confirmation. You look at the pattern over here and then look at the transfected cells. So it helps in detecting conformational antibodies which are otherwise missed on the line immunoassays and the ELISAs. So that is what we are currently been using all across. But then, now there is a lot more to come. There are new emerging technologies which are available, which will be able to hopefully overcome, and with so many new autoantibodies being detected, it might be required that we need newer technology, and that is multiplexing should be possible, it should be objective, it should be giving report in quickly, within hours, numerical results, and we should be able to standardize. So, but are we still there? Let's just look at it. So here there are something called as addressable laser bead immunoassays, where we use multiplex beads of different colors, each color attached to a different antigen, and then measure it under the Luminex. It enables rapid, economical, quantitative detection of about 50 to 100 antigens in a sample. Then there is one more technology which has come, which is the cytobead assay. So what you do here is, it integrates the Hep2 of ours along with the specific antibody. What we do currently now is, do an ANA, if it is positive in a tiered way, you go for ANA positivity, then you look for the other specific antibodies by either ELISA or a blot. Of course, there are some areas, even if it is negative clinical symptoms are there, you have to be interpreting, but otherwise, a general statement. So here, but then they try to see why not we bring them two together. And instead of doing first an IFA and then doing an ELISA or a so these are the slides which are available. Central area has the Hep2 cells, and here you have the different beads which are there attached, and you are able to identify different antibodies. So this is what we are actually seeing over here. Can you see this? And this is for also for ANCA this has come, and as well as for some of the celiac disease cases. So where you are able to see that you do an IFA also as well as the bead assay. So these are the assays. They are still in pipeline, some of them. They are not really into diagnostic use, but they have been tested on a few diseases particularly, but not all antibodies what we are currently are there on that platform. Then came the antigen microarray technology. This definitely made a huge difference. You have microarray plates, microarray assays where you can detect about 48 antigens, 50 antigens which are there, and they allow simultaneous analysis of antibody against a large number of antigens. And not just that, we can also see that you don't have to do one sample at a time, you can do multiple sample processing at a time. You have two techniques. One is the planar, where you use a slide on that involves immobilizing of the antigens, and the other one is the bead. Bead is definitely having higher multiplexing capacity, higher of sensitivity. So this has really changed because we were able to stratify the different lupus patients, the dsDNA antibody, the SM antibody along with the patient. You know that yes, this patient is going to be likelihood. So they could do a biomarker, actually a study of the entire SLE antibodies using the microarray, and it provides a very comprehensive view of the autoantibodies. The biggest challenge now currently is they are very high cost. But slowly costs are coming down, and it may be possible that you will be able to use it in the near future. This is a very smart technique where they have done, what they have done is, they have taken the gene, recombinant gene from a jellyfish which fluoresces, which gives light, to which antibody has been tagged, and then they add other antibodies where you attach the protein and look for the presence, measure it under the luminometer. So this is particularly useful for identifying new novel antigens, and this has been useful but currently not really into the diagnostic. The advantage of this is, as I said again, like how you use in cell-based assay, here they use crude extracts in the COS1 cells, which are the Renuka luciferase antigen which is being used, that is the RUC, and it doesn't show, it doesn't go the loss of the epitopes during the purification process, that is retained. So that is the major importance of this particular assay. And this is one, I think after this I will not, where you have used, samples are exposed to used cells with genetically barcoded, and they have 2,688 distinct antigens. So it's a technique for comprehensive and high throughput discovery. The entire X proteome can be put on that, and you are targeting the antigens over here. And you again do the analysis by an advanced immunophenotyping technique, but again mainly useful for discovering novel diagnostic antibodies and mainly in the field of autoimmunity research. So we are really, it's going to be not very far that we are going to see even more antibodies which are coming. How is it that we are being detecting? So just a small brief. So currently Albia, that is the laser bead immunoassay, which is already been available, some equipments are there and they can be put into a clinical setting. Then cytobead assay is already available, though not regularly in our country. Then there is a likelihood that luciferase immunoassay may be possible, but where we mainly useful for epitope mapping rather than for doing the regular work, mainly for research. Microarrays has definitely having those chips and scanners which will make it as a good useful thing for our future for autoantibody analysis, and we will be able to do the biomarker panels for these patients, take a case, work up on the entire antibody, decide how will this patient perform by the help of this thing. And here REAP, it is yeast display, again you require a NGS or a FACS sorter, but it has mainly for research purpose. So these are just, if I wanted, I know this sounds so many names and so many techniques, but basically it is to say that just to, when we started 20 years back and I used to tell about some of the autoantibodies that we use, they used to seem so difficult, and now with regular use we are all so aware of this, and it's not going to be very far that these will also come in. What are the standardization challenges? We have already said is the antibody variability, epitope recognition, reference standards. WHO gives us reference standards so that based on that the traceability is done, but then there is a limitation of reproducibility from patient sera, interassay variations because of across different platforms. It has been very difficult to standardize in spite of various efforts which have been taken by the WHO as well as the AAC, which is working on this for this. So what is best way now? Currently what they are doing is, even if you can't standardize, at least do the harmonization. So how do you harmonize? Because each patient gives you, lab gives you a different result. How so what do you bring in to bring in uniformity in reporting? And that is what we are doing, aligning the differences between different platforms, whether it be a ELISA, a blot, or whatever. How do you do? First of all, when it comes to and for testing, because it is important that the patient should get the right diagnosis without having unnecessary false positives, unnecessary workups. So when do you do? There are defined indications when to test based on the clinical manifestations. So that brings in one harmonization. Testing algorithms. ACR ULR has actually now given a tiered approach. Recently I just saw that article wherein you can actually say if this antibody is positive, then what do you work up? If I see a homogeneous pattern, what should be the antibodies I should do? If I see a nuclear pattern, I should work up on the myositis antibodies. So they have actually given a tiered approach for it. Reporting guidelines, there is a lot of effort which is going in for reporting of ANA, for instance, which is, as I said, the ICAP nomenclature for ANA on reporting of H2, HEP2 and the likelihood ratios. So it would be good if you could give the reports with the likelihood ratios. There is an effort on that. Right now it is not happening. What is the clinical disease association and what could be the likelihood that this patient will have? So this is one way of working, and how we can probably do is AI integration, wherein automated, of course, AI integration has definitely helped us for the ANA reporting to some extent, automated pattern recognition, trying to bring in standardization across, enhanced predictive modeling. Suppose now a patient has anti-dsDNA, anti-C1q, HLA genotype, proteinuria 86, there is high chance that this patient is going to have renal involvement. Multimodal analysis, data integration, personalized medicine, serotype guided therapy. Depending on what autoantibody you have, you treat the patient. Not that for all SLE cases you will treat in the same way. Whom will you treat with B cell antibodies, anti-B cell therapy? Where will you use which? There would be a serotype guided therapeutic and novel biomarkers, of course, which help in microarrays, and the LIPS technology will help us. So currently, as far as the key takeaways for today is, IF remains the gold standard when it comes to the detection of ANA, and when it comes to ELISA and line immunoassays, they are useful for specific antibodies when you are detecting further, whether it is a dsDNA antibody, a systemic sclerosis, whether you are using Me2 antibodies or whatever it would be. Line immunoassays and ELISAs will help. Emerging technologies improve the detection of rare, and we will get more antibodies in the future. Harmonization is of course very critical. Quality assurance is of course goes without saying, it is very critical. Disease specific algorithms are required to optimize the cost, and AI integration will definitely revolutionize the pattern. So where we are also heading, other things which are coming in the pipeline is the point of care instrument. So there is a chance that patient comes with joint systems, you do a point of care comprehensive antibody panel which will be available in 15 days. AI will help us further if they are positive to do for HLA genotype, NGS, antibody panel, clinical data, do a system output, look at the confidence diagnosis, organ involvement, which organ will be involved in this particular case. Targeted therapy also, CAR T cell and CRISPR based treatment designed for specifics and monitoring this variable. We are already using CGMs for our glucose. I think I'm wearing one now, but yes, so that is what I'm trying to say. So that the day is not too far. I'm not saying it might happen in the next 5-10 years, definitely not in the next 15 years, but it may be possible. So what I would like to say is, if you are a clinician, order smart. Look at what are the indications, when do you want to do the test, just don't unnecessarily write a test. Interpret with context of your clinical features, and refer for novel antibodies testing when you are suspecting a seronegative case, not for all, because the challenge is, it is very clear where you have autoantibody positive, but there are so many seronegative cases and still the patient has strong clinical features. Look for novel antibodies for these cases. If you are in the lab, master the methods. We need to be updated. Stay updated on the harmonization guidelines and prepare for AI integration, otherwise we'll be left out. And if you are a researcher, of course, it is a window into disease mechanism and therapeutic. So for this immunology, this is one of the immunology's most rapidly advancing fields, and continuous learning is essential when it comes to autoimmune diagnostics. Thank you so much.